Good afternoon, everybody, and welcome to today's Congressional Biomedical Research Caucus briefing. I'm glad to see a full room. We might have more sandwiches coming for those of you who don't like ham sandwiches. That seems to be the only thing left, so we have a few more things coming up. I would like to start by thanking the Congressional Biomedical Research co-chairs because without them, these events could not happen. And those co-chairs are Representative Steve Stivers from Ohio, Representative Charlie Dent from Pennsylvania, Representative Jackie Spear from California, and Representative Rush Holt from New Jersey. It's truly because of their commitment, dedication, and ongoing support that we're able to host these caucuses. Today, we will be hearing from Dr. Sean Brady from Rockefeller University in New York City. Dr. Brady is a chemist whose research has him sifting the soil for treasure. And the treasure he is seeking is a source of new antibiotics. And we all know we need new antibiotics. Today, Dr. Brady will discuss this relatively new area of research and the promises and challenges it holds. Dr. Brady graduated with a degree in molecular biology in 1993 from Pomona College. He received his PhD in organic chemistry in 2001 from Cornell University. In 2002, he moved to Harvard Medical School as a fellow in the Institute of Chemistry and Cell Biology. He was named an instructor in the Department of Biological Chemistry and Molecular Pharmacology at Harvard Medical School in 2004 and moved to Rockefeller as an assistant professor in 2006. He was then promoted to associate professor in 2012. Dr. Brady has earned many awards. And among them, he has a Howard Hughes Medical Institute Early Career Scientist Award, which was given to him in 2009. I'd go through all his awards, but I think I'm going to stumble over some of the words, so I will leave that at that, and it is my pleasure to welcome Dr. Brady here today.
The ultimate thing I want to tell you or convince you is that we're developing processes by which we can really start from dirt, extract DNA, and use that DNA to generate a new family of antibiotics. But before we get to that process, let's talk about the traditional way in which antibiotics were derived. And essentially, the majority of anti-infective agents and the majority of anti-cancer agents that are FDA approved come from natural sources. So, okay, they come from extracts of these things. So you can start with plants, you can start with fungi, you can start from bacteria, you can start from marine sponges, you can start from amphibians, you can start from insects. You grind them up, you take those extracts, you ask whether they have activity as anti-cancer agents. Do they kill human cancer cell lines? Do they kill bacteria? Do they kill fungi? Right, that simple approach leads to the majority of clinically relevant compounds we know in those fields. And there are just some examples you might know something about. So here's vancomycin. It's the antibiotic of last resort. Right, it's given to you in the hospital if you have methicillin-resistant staph, or otherwise known as MRSA, which you see often reported in the popular press as one of the major threats to hospital-acquired infections in the U.S. today. So that compound is used in the clinic in its exact form as it's produced by a bacterium. No modifications by a synthetic chemist. It's used the exact same way it's actually produced by a Streptomyces. So here's rifamycin. That's the lead structure for the development of lots of anti-tuberculosis drugs. Here's tetracycline, broadly used antibiotic, used for a number of years. Here's streptomycin, another antibiotic used for a large number of years. Not only do we get antibiotics, but we can get anti-cancer agents. So here's staurosporin and apothelone. Those are lead structures for the development of a whole range of now anti-cancer agents. Here's rapamycin. It's an immunosuppressant. So lead structure for these developing ideas for how you can do organ transplants and have immunosuppression. So this slide has probably been up there long enough that a large part of this audience is a little bit nervous. They're going to have to hear about this the whole time. Brings back some kind of horrors of sophomore organic chemistry, right? And to some extent that's true, but actually I don't care for this talk that you understand all of the details of that. What the point of putting that up there is to tell you that they're different. And if you want to go through my entire talk and assign a fruit to every structure, that's perfectly fine with me. Right? What you can't do through the talk, you won't follow what we're doing, is if you say they're all the same. That's apple, 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 right? We take the time to draw those structures for really two key reasons that are going to be important as we go through this. That is that very small differences, whether you have an orange or whether you have a lemon, right, actually are tremendously important in drug discovery. Right? A single atom change in one of these structures can generate an antibiotic that's better or completely obligate or abrogate the activity of that molecule. That's one of the reasons we draw the structures. The other we draw the structures is to tell you that we found something new. Really, one of the driving forces that we're going to think about is how do we find new compounds? How do we find lime nuts? How do we find fruits that you've never heard of? Right? So the two key points to take away from this slide, and it doesn't matter after this that you actually understand the structures, is that small details are pretty important in drug discovery. And the ultimate goal of finding new antibiotics is looking for things that we've never seen before. How do we develop strategies to do those two things? So I'm going to draw them like this. There's one common theme amongst all of those compounds. Does anyone have any idea what it is?
They're all organic.
They're all organic. It's one. They don't all have the same activity. They don't have the same structure. They all come from bacteria. Every single one of those drugs was isolated from the culture broth of a bacterium. So as much as biology in general has been a great source for biologically active small molecules, bacteria has been the leading source for anti-cancer agents, antibiotics, and antifungals, essentially cytotoxins. And the process by which we find all of those is actually traditionally very simple. It largely hasn't changed since Fleming makes that discovery, that one microbe can kill another, and leads to the discovery of penicillin almost a century ago. We've been using the exact same process for finding drugs since he made that observation, and it's outlined here. Really simple. You go to the field, you collect an environmental sample. In many cases, that's dirt, because dirt is one of the most microbial rich environments on Earth. So you go into dirt, you culture what you can out of it, then those organisms grown in pure culture, the starting point for the discovery of new antibiotics. Grow them in fermentation flasks, and ask which fermentations will kill bacteria. If you find one that does, isolate that compound, and lo and behold, you get a new antibiotic. Very simple paradigm that today a high school student can do. Right? It leads for the first 30 or 40 years of doing it to tremendous discoveries in antibiotics. Almost every antibiotic you probably know the name of was found pre-1965. That's the golden age of antibiotics. And then we have what we're now calling the innovation gap. After 1965, essentially there's this absence of antibiotics coming onto the market. There's a few very recently that have come on the market. They were actually discovered largely back here, and they're just being introduced now. So we have no antibiotics coming on market. The second bad thing about that is you can look at every single one of those compounds now has resistance in the clinic. So one of the take-homes from that type of slide is that maybe we've run out of antibiotics to find in nature. Right? It wasn't that people stopped looking for them, they stopped finding them. Right? And the idea here is that people looked and looked for antibiotics, they just didn't find any new ones. Well, we now think that's somewhat misguided, and we think it's misguided because of what we now know about the limitations of that approach. If you think about that approach in some detail, there are actually a number of limitations. I'll talk about one today, and that is if we look at that soil sample in real detail, we don't just find the 100 or so bacteria that you traditionally grow out of it that have led into the pipeline for drug discovery. If you look at it in detail with modern sequencing technology and sequence very deeply into the DNA you get out of that sample, there's suggestions that a single gram of dirt can have 10,000 different types of bacteria in it, 99% of which we never grow in the lab. We can't grow them, we can't get at their chemistry. Maybe there's lots of new antibiotics that we haven't discovered. And the issue isn't that there aren't antibiotics, it's just we've been scratching the world in the same way for 100 years. Right? We need to find a new way to scratch biodiversity to find molecules. So just to give you a measure of size of uncultured versus cultured, that's 3,000 jelly beans. It's a pretty good representation of the number of different bacterial genomes thought to be present in a single gram of dirt. Go into your backyard, dig up a spoonful of dirt, there'll be 3,000 to 10,000 different types of bacteria in it. What we traditionally look at is about 1% of those. Everything we know about antibiotics, essentially from bacteria, comes from looking at that group of organisms. How do we develop methodologies that allow us to look at compounds that are produced by that large group that's out in the environment that we haven't been able to look at using a traditional drug discovery paradigm? So the next couple of slides are a little bit of detailed science, but you have to understand it to understand what I'm going to tell you about. And in order to make a small molecule, right, like an antibiotic, they're relatively complex structures. They require a large number of genes to encode their biosynthesis. Okay, so you need genes to make molecules, you need genes to regulate the expression of those other genes, and you need genes to confer resistance. So how does a bacterium make an antibiotic without killing itself? It has a set of genes that confers resistance to that antibiotic. It turns out that all of those genes in a bacterium are clustered together on the bacterial chromosome. They're not randomly distributed throughout the chromosome. What it means is you can take genomic DNA from a cultured bacterium. You can chop it up into pieces. If you leave those pieces sufficiently large, some of them will have entire biosynthetic gene clusters on them. You introduce that into model systems in the lab, they now have all of the genetic information to encode that molecule and actually often gain the ability to make those original molecules. So you can redraw the drug discovery paradigm to look something like this, where not only do we get molecules out of the organisms that actually make them that you get natively out of the environment, but you can take DNA from these organisms, you can introduce it into model systems in the lab, call that a gene library, and you can look at individual members of that library and identify clones that are making those same compounds. Right, so how do you get molecules out of this group of organisms? Well, it's pretty obvious now, right? You skip the step you don't know how to do. Instead of isolating DNA here, we'll isolate DNA directly from the environment. It's called environmental DNA. The modern term for it is metagenomic DNA. We take that DNA, we introduce it into model systems in the lab, and we look for clones that are producing new chemistries. An orthogonal set of chemistries that we saw there. If you're back to this, there's your lime nut, right? That's a different fruit than we've seen in the past. There's a couple of steps that one has to develop to figure out how to do this. One is, how do you get DNA out of dirt? You can probably start to imagine how you might get DNA out of dirt. There's lots of ways of doing it. The simplest one is just take dirt and heat it in the presence of a detergent. We don't actually use Tide, but we use other detergents in the lab, right? And that heating bacteria in the presence of a detergent pops them. It lyses them. That releases their DNA. You can spin out the mud, and from the supernatant, you can precipitate DNA. And that's precipitated DNA from the environment. So it's not quite pure. It's mud, but it's mud highly enriched in DNA from all of these organisms, or lots of these organisms we haven't been able to grow in the past. We can then purify that DNA, and that's pure DNA here. And once it's pure, that's no different than DNA you would get from any other organism. So DNA is DNA. You can take standard, well-described processes for introducing that into model systems in the lab. We can then go from an environmental sample to a large environmental DNA library in somewhere between a couple of weeks to a couple of months, depending on how many millions of clones you want to capture. So the problem is not generating these large libraries. It's how do you identify the rare clone in there that's actually making an antibiotic. So let's just look at some real-world examples. We have now collected soil samples throughout the United States. We've extracted DNA from them, and we've generated libraries from them. We call them mega libraries. That is, they're the first libraries that are large enough to begin to capture the true genetic diversity that's present in a soil sample. And you need to do that so you can recover large gene clusters that might give you molecules. So just to give you a matter of size one more time, how big is that library? Well, it requires about 20 million clones to cover all of the microbial diversity that's out there. You could take that library and you could take a genome from every different type of large animal in Africa and you could stuff it in that library. Or the equivalent size is taking the genetic diversity out of about a kilo of dirt. What's that meant to say? It's meant to tell you there's a tremendous amount of genetic diversity out in the environment that we just haven't had access to in the past. And now we have it stored away in the lab and we can begin to look at it. We look at it in a couple of different ways to try and find antibiotics. I think the conceptually easiest one is just look for activities. I played out that library, played it nice spatially arrayed so each one of these colonies is an individual clone, and let it grow up. You immediately see something's happened because some of those clones turn colored. Right? You get blue clones, you get purple clones, you get brown clones, you get yellow clones. That's a result of the DNA you've cloned in there. We're not really interested in colors, but that tells you you've cloned something. We're really interested in antibiotics. How do you find antibiotics? Simply overlay that with a bacterium you want to kill. In most cases, you get a nice lawn growing up. Every once in a while, you'll see a clone where the bacterium can't grow around it. That's producing an antibiotic. Right? We simply pick that. We treat it just like it's a cultured bacterium now. We grow it up. We ask, is it making small molecules that will kill other bacteria? We can characterize those, and you get a small collection of new antibiotics. Everything shown in red is a compound that hadn't been previously described from cultured-based studies. So that seems like a nice, easy way in which you can find lots of antibiotics. And in reality, it's easy for the graduate students and the postdocs that are doing the work, let's say. But there's one person it's not particularly easy for, and that's for the E. coli, or the model system in the lab that you're actually asking to do all of the work. Right? You start from this soil sample, which contains 10,000 different types of bacteria. You isolate DNA from it. That's this extraordinarily heterogeneous population of DNA. Right? You introduce it into a single model system, which is E. coli. That's this great genetic workhorse that all microbiologists work with. You introduce it in there, and you ask it to take genes from the environment and convert them to molecules. It's a long ways from genes to molecules, right? I mean, you have to go through RNA, you have to go through RNA, you have to go through proteins, they have to find their substrates, they have to build them up to molecules. And what happens is E. coli goes awry on almost all pathways. It doesn't know how to deconvolute that information into a molecule. It does in some cases, but there are lots of pathways captured in these libraries that we're not turning on. So the question was, how does one turn on more of the pathways so you can get a more diverse set of antibiotics out of these samples? And you go back and you say, how does nature do it? Well, you got that DNA from lots of different bugs. Why not introduce it into lots of different bugs? And so one of the things my lab's been doing is developing new models that we can introduce environmental DNA into. So instead of just using the standard model, we've developed a variety of other bacteria that we can now introduce large libraries into. We can repeat the same screen and look for antibiotics being produced there. And essentially every host we choose, we can find new antibacterial active clones. And now what happens? What's the chemistry being produced here and here? Well, it turns out the chemistry being produced in different bugs is almost always different than the chemistry you found in E. coli. By taking the same library and putting it into a different background, you get a completely different set of genes turned on, and as a result, you get a completely different set of antibiotics. All right, and so one of the ways we can find molecules is simply move these libraries into more and more diverse hosts. And one of the things we're working on is how do we move them into more and more diverse bacteria so you can get more and more of the genes turned on. So that's one way you can find antibiotics. We can kind of look at the flip side of that. That's the end product. Let's look at the very beginning. Can we do a gene-based search that might allow us to find gene clusters that make antibiotics? Instead of looking for the product, let's look for the actual information that encodes that product. So we go back to our gene clusters. We know that gene clusters encode molecules. It's also true that these compounds that have found their way into the clinic are often not the only version of that compound that's ever been found. Lots of vancomycin-like molecules have been found from nature. This just happens to be the best one that's been found to date. Lots of streptomycin-like compounds have been found, but that's the best one that's been found to date. They've come from screening 1% of the bacteria. Are there lots of better ones out there that we just haven't found? And how do we go about doing that? Well, we're going to do that by picking a gene in these gene clusters that we think tags to this biosynthesis, and using it as a hook to go out and find more gene clusters that might make new versions of those molecules that could be better than the ones we've seen in the past. So here's an example of doing that. We're doing it with what are called the antibiotics of last resort. So chemically they're called the glycopeptides, but that's vancomycin and teicoplanin. Has anyone ever heard of either of those? Yeah, okay, so these are the antibiotics of last resort. They're given to you in the hospital if you have methicillin-resistant staph. For lots of cases, that's the final line. You don't respond to those, well, you know where that goes. So these are the compounds. About 200 of these were found. They were the major kind of target for large pharma for about 40 years because they were the last line of defense. They go out to cultured bacteria. They find 200 derivatives. These are the two that make it in the clinic. Our question is there a large reservoir out there of these that people have missed in culture-based screening that might give you new versions of these? So let's just see what's going on here. We'll cartoon it so we don't actually have to look at the structure. The whole family of molecules basically has a common center, and they just differ by the detailed chemistry going on around the edges. And that detailed chemistry, which I've just colored in the more detailed picture, dictates whether you're a good antibiotic, not a good antibiotic, and in some cases whether you're active against drug-resistant bugs or not active against drug-resistant bugs. So can we find molecules that look like this, that have new derivatization patterns that maybe are better than the ones we've seen in the past? Well, we need a gene, so we pick one gene out of the biosynthesis of these that we're going to probe the library for. And we thought that gene was the most unique gene in the biosynthesis of these. It happens to make that bond. We call it an enzyme that makes that bond. So that's exactly what we do. We collect soil, we make a library, we probe it for this gene, and we recover just that subset of clones that have that gene on it. So now we've whittled down our 10 million clones to a handful of clones. We sequence those, and lo and behold, we find biosynthetic pathways. Where here's our gene of interest. It's coupled to biosynthetic pathways in every case. And if you compare those pathways to ones we know make this family of molecules, they look very similar. It doesn't take a PhD in bioinformatics to say these are similar to those. Same gene organization, largely same gene content. And it suggests that these actually might make new derivatives of that family of compounds. Right, there's one big difference, and it's right here where you see the rainbow of different gene colors. Each one of those different colors indicates a different function. And those are the genes that encode enzymes that do the functionalization around the molecule. So essentially, in every case we look at from the environment, we find a new functionalization pattern. So what have we done so far? We've taken dirt, we've extracted DNA, we've made a big library, we've culled it down just to a subset of biosynthetic pathways that we think have features that indicate they might make antibiotics of last resort. The details suggest that's actually going to be the case. How do we get molecules? Well, we just introduce these into model systems in the lab and begin to see what chemistry is being produced. And lo and behold, we get a whole new collection of glycopeptides. All right? It's not always you put the whole pathway, and there's lots of different ways you can do this, but the basic idea is you take those genes, you put them in a cultured bug, and you see what you get. Everything shown in red is, in fact, a new functionalization pattern that hadn't been seen from cultured bacteria. I can almost guarantee you that if you went into your backyard, if you have a backyard here, and dig up dirt, and you take this process, you will find new glycopeptides in there. That's how common these are. Every single soil sample we've looked at around the world, there are indications that there are additional glycopeptides derivatives in there, that we find new sequences of these families suggesting there's new gene clusters. Huge reservoir of these out in the environment that we just haven't found yet. And at least these compounds, the ones on the left that are highly sulfated, have activity against some clinically relevant drug-resistant bacteria. So that's our lime nut, if you will. All right, one last story. So the question now is, how do we find brand new things? So I've told you that we can go out and we can take a gene-based search for finding new derivatives of lots of different antibiotics. And I just gave you one example. So let's take the exact same type of search where we're going to look for a gene that we're pretty sure is involved in making new antibiotics, right, or making antibiotics. And instead of looking for sequences that are very closely related to things that make these guys, let's specifically look at the sequences that are as different from anything as we've seen in the past and ask whether they make brand new chemistries. So here's the process. We collect dirt, we make a library, and we begin to sequence in that library for specific genes that are involved in making natural antibiotics. And this is one way in which you can organize genes. So each one of these lines is representative of a different gene. And if it sits next to something else, it means they're closely related in sequence. If it's far away, it means they're distantly related. So now they're organized in a way you can begin to think whether there are relationships between known sequences. And what I've told you in the past is let's look at things that are related to tetracycline and find new tetracycline derivatives. Let's do the exact opposite. Let's stay away from all of those and just pick out sequences that are as distant to anything we've seen before and ask whether they generate brand new chemistries. So that's what we do. We then recover those clones. We introduce them into a model system in the lab. And immediately you can see something's happening. Where here's the model system without any DNA in it. Here's the model system with DNA. There's brown, there's orange, there's purple. So clearly something's going on. Lots of small molecules are colored. It's a nice surrogate to say something's actually happening. To make a long story short, we go through and characterize all of the molecules being produced here. And that's the collection of compounds that come out. Every single one of those is different than a molecule that's been found previously from a culture-based study. And that's pretty good, because if you go back to our kind of timeline slide where we have this gap of innovation, if you go to large pharma and you ask them what happens if you screen for compounds from cultured bacteria, they'll tell you 99 times out of 100 we find the exact same compounds we've seen before. Right? And at least it surely looks like here we're finding something that's different. How new are they? Well, some of them are nominally new, some of them are actually pretty significantly new. One of the ways we judge as a chemist whether something's new or not is you strip away all of the details and just look at the carbon skeletons, the ring systems that are there that the compounds are based on. That's a brand new ring system. That's a brand new ring system. That's a new carbon skeleton. That's a ring system that's been seen. That's a ring system that's been seen. But in each case, it had only been seen once previously in culture-based studies. So it sure looks like if you can drive your discovery by bioinformatics, you can find whole new collections of natural products out there in the environment. This is now our large collection of lime nuts that we can test to see whether they have new activities. Right? So this is exactly what we're doing here. We put a compound on a disk, and we then let the bacteria grow up in the background, and we look for compounds that have zones in the growth inhibition, indicating that they're producing antibacterial activities. Right? And that's this one so far is the most active. We've called it the terramycin A because it comes from Arizona, and it has a tetracycline ring system. It's active against methicillin-resistant staph. It has a completely new carbon skeleton, never been seen before. Suggests that we can in fact get at not only derivatives of known things, but absolutely brand new antibiotics using that strategy. So I'll finish up with the last three slides. What are we doing in the future? Well, we're trying to scale this, right? We're collecting literally hundreds of samples throughout the United States. This just shows collecting in Arizona, New Mexico, because they're third and fourth most biodiverse states in the country. Collecting in New England, because it's way different from Arizona and New Mexico. Right? Isolate DNA, sequence that DNA, populate it into computers, and we've written software that now allows us to compare all of that sequencing data, not to individual molecules like I've been doing, not just looking for glycopeptides, but compared to every biosynthetic system that's known. So you can look for any family of compounds you want now. And you essentially are generating maps that allow you to say, where are you going to go in the future to find molecules? Where do we go to find more glycopeptides? Well, it tells you want to go to all three of those sites. Where do we go to find these anti-cancer agents? Well, you want to go to Texas. Where do you go to find new immunosuppressants? Well, you want to go to that California sample. Not only does it tell you that, but it also allows you to, where are the most diverse environments in the country so you can go find brand new things. Right? I think it's a reasonable analogy to say that we're not exactly Google, called it eSNAP. We have postdocs and graduate students driving around the country, collecting soil samples, extracting DNA, and mapping this biodiversity, which I would argue is a significant change in the approach in which you find molecules than what has been done in the past on this Fleming-based discovery strategy where it's completely random in the past. And I'll finish here. This is just a sampling of other molecules we found using that strategy. We're not only finding antibiotics, we're really focusing on anti-cancer agents at this point, and we at least have some where I can't show you the whole structures, otherwise we can't patent it, but that molecule is more active than anything known in that family, and it's active against drug-resistant cancer cell lines. Right? So now we can go out and make direct searches for anti-cancers as well. Here are the people that actually did the work. Jeff Craig did the early work on the phenotypic-based screening. He was a graduate student. Jacob did the glycopeptide work. Ziyang Fang is a Chinese postdoc who did the aromatic polyketide work. And Jeremy Owen is a postdoc from New Zealand who did all of the bioinformatics at the end. The funding comes largely from NIH. That's why I'm here, I guess. And then Rockefeller University and the Howard Hughes Medical Institute. I'm willing to take questions. Thanks for your time.
So you kind of touched on it in the end where you were talking about the sequencing. I'm wondering how like sequencing or deep sequencing would compare to your original kind of DNA extraction? Are you picking up rare species that might be in the soil?
Oh, so yeah, by deep sequencing, we see a huge number of things out there, right? I mean,
Yeah,
so we.
that's not being picked up in the initial.
Well, I mean, we think our libraries are big enough that there's a certain cutoff, right? We're not going to see extraordinarily rare things, but our libraries are big enough that if we find a sequence, we can almost always find it twice. So the major things are in there at some level where you've actually saturated them. But I think all environments we're now understanding that there's then a rare population that by cloning it's going to be hard to get at, right? So there's no question about that. I mean, if we go through and we do sequencing, so if we go back and try to talk about this in some detail, what do we learn from that? Well, I can tell you that in a single handful of dirt, you can find 10,000. Well, where is this? So these are just two different, so there's generally a line down this slide. These are known as polyketides, so it's one type of biosynthetic system. These are known as non-ribosomal peptides. So they're made from amino acids. These are made like fatty acids. And so we screen them separately. We can find 10,000 different representatives of these in a single gram of soil. We can find about 2,000 to 5,000 of these, all hidden in a little gram of soil when we start doing sequencing. Tremendous amount of biosynthetic diversity. And then one of the things we're interested in doing is we're collecting all these samples, not just uploading them, we're doing a lot with that data, right? I mean, how do samples differ from one sample to the next, right? Is it worth going out and collecting hundreds of samples? And it turns out that samples largely have about 1% to 2% overlap sequence-wise. So not only are samples diverse, but they're different from each other. And it turns out that if anyone's here from the state of Arizona or New Mexico, no? Yeah. So it turns out desert samples are way more diverse than non-desert samples, which is something I think people are beginning to look at in other ways, but as far as chemistry goes, no one had looked at that. And they're significantly more diverse. And so that's why if you look at our pictures of where we're making libraries, lots of them come from those areas. So Arizona has been diverse from the dawn of ages. Yeah.
What share of your work is dedicated towards extracting out after finding a character or common characteristic versus trying to find widely diverse characteristics?
Yeah, so initially, if you want to see the history of my program, right, we were just going to look at brand new things, right? And that just ends up being a really high bar. We have to develop lots of technologies. And so then you go after things that look like they would have high rates of return, like finding new derivatives of clinically relevant compounds. So we spend a lot of time on that now. And in the background, we just don't publish a lot on it because there's a lot of stuff to be done there to try and develop that as a technology. So I think finding new derivatives is a set in stone thing. We can do that now, right? And we're just coming online with getting all the things together to really scale the idea of finding brand new things. Is that the question? Yeah. So the whole field, the field's been around for about 10, I guess 12 years now, was set on finding absolutely brand new things. And the whole field was set on that very first story I told you, that we would just dump DNA into bugs, and there would be these gobs of antibiotics heading our way, right? And in theory, that's nice, but it turns out there's lots of technology we have to develop. How do you get DNA out of dirt? How do you clone it? What bugs are you going to use? How do you screen it? All those kinds of things. And so yeah, lots of my lab is involved in that basic metagenomic science. How do we develop that technology? And we're getting there. But it's not quite turn crank yet. I would say finding new derivatives is getting to the point of where we can turn the crank and find new things that way.
As a basic scientist, can you give us an idea of how, I guess how you're going through the process of the what next? So you're finding all this fantastic stuff. Obviously, we know that there's a huge gap in the drug development pathway. So how are you traversing to try to get these actually to a point where it's feasible?
Well, so that's the question. Are we taking molecules beyond the point I've just told you about, right? And that's a tough one, right? And it's something I honestly think about as to what we're doing. I mean, is it better as an academic to develop the technology that'll teach people how to fish, right? Or is it better to fish? And I would say we're now walking a fine line between both of them. That we will take our best compounds and try and move them forward, right? As opposed to in the past, I would have said we wouldn't do that. But we would just develop technology. We would try and develop methodologies that would allow people to do environmental DNA research. I think in the real world setting of NIH and the real world setting of science, you probably can't survive doing just that. And so we're now taking our best compounds and thinking about how to move them forward. And I don't know the answer. It's something that's new to me, right? But we are switching the group from just developing methodologies for environmental genomics or environmental DNA strategies to what's our best compounds, what do we do with them next? And
So what's your approach? Are you targeting pharmas? Are you trying to do this yourself? Like how?
yeah, so my own approach will be to try and do it myself. Yeah. And so I mean, whether I mean, again, we're wading into that, right? I mean, but there is a renewed interest. So I think maybe I'm somewhat lucky because there is somewhat of a renewed interest in this field, right? That as one realizes, it died. I mean, natural products died. Merck closed their programs, Pfizer closed their programs, Wyeth closed their programs. There were no industrial natural product screening programs left 5 years ago. And if there were, they were very small. Right? And so there were no startups doing any of that either because no one could find any new molecules. Now the idea that if we take DNA-based approaches, there's all these new molecules, there is actually this kind of bubbling of interest out there where we think we may be able to do it in a venture setting. But I don't know, right? I mean, I would say to some extent we're just lucky if we get to do that because it's the right timing, right? It just happens to be people are interested in these ideas at the moment. If you just had a molecule from a high-throughput screen, right? I don't know what you'd do. I mean, to be honest with you. I mean, some of that would just hope that someone comes and licenses it from a university.
And it's just such an interesting problem. You know, the whole concept of the valley of death with drug development is, I mean, again, you're turning up fantastic science, but actually making that
Is tough.
getting it to people who are currently dying from MRSA isn't really.
Right. So we'll see. We have some other compounds we really like, and we're starting down that road. I mean, it involves lots of things. How do you get enough compound to do that? It doesn't just cause, I mean, it's money, but it's also resources of how do you generate enough compound? Are these bugs making, how do you engineer them to make more compounds so you can have enough to work? And those are kind of things that are interesting to me, right? I mean, we're willing to do that. But the actual getting enough to put in mice and the people, you have to convince someone that it's interesting. And yeah, you give it to your university and they go out and find someone who's interested. I mean, that's what it comes down to, right, at the end of the day.
What makes the deserts more diverse than other areas?
We don't know. I mean, I would love to answer. You could say that maybe there are different microenvironments there, right? That there are lots of different microenvironments as opposed to an organic rich soil where all the soil looks largely the same. Seems logical to me. I don't know that that's the answer. We know that there's more microbial diversity of the families of bacteria that traditionally make molecules there. We don't know why that is. You can come up with lots of strange rationalizations, but I mean, coupled to this type of work is us looking in detail, so the whole nother talk on looking at the individual soils that constitute these and asking, are there parameters in soils that would drive that diversity? And so far we don't find anything that really drives a single factor that would say, look at this factor and there's going to be lots of diversity there. So it's going to be some complex idea that more microbes can go there because there's more microenvironments? I don't know.
My question is specific to MRSA. As a predominantly field-supported infection, I was wondering if it would be more advantageous to study the pathogenesis to devise more of a preventative vaccination as opposed to an antibiotic. Is that something that's being considered?
Sure. So there's a whole other arm of my group who studies pathogenesis, and our niche in that field is that we like to try and understand the small molecules that play a role in pathogenesis, and do they represent new antibiotic targets. So we don't look at MRSA because the whole world looks at MRSA, right? And kind of enjoy looking at things that there's not so much competition in. So we look at opportunistic pathogens, things that will become important as people get older, as diabetes becomes more popular. I mean, as it becomes more prevalent, as more transplants, as age patients survive longer. All those opportunistic pathogens are becoming real serious problems for which we don't have drugs for. And so that's the niche we're working on. With that idea, can we stop signaling systems in those bugs, right, that would then stop them from being pathogens, or stop them from producing toxins that would stop them from being pathogens? So yeah, that's part of what we do, centered around small molecules.
You mentioned that during the innovation gap, it wasn't that people stopped looking, it was that they stopped finding things. Did people not try to develop new techniques that they're using? Are you the first deviation from Fleming's techniques?
Well, I mean, I'm not going to say I was the first at anything. but I mean, yeah, this generation is the first that's really using DNA-based strategies to drive drug discovery in the microbial world, right? It's taking advantage of the DNA-based technologies we had. What were the strategies people did in the past? Well, they struggled really hard to get new bacteria growing, right? So they used strategies, they just weren't these strategies, right? And they worked really hard on so there was actually a bump up in at least anti-cancer discovery, anti-cancer molecule discovery, when people moved from terrestrial natural products to marine natural products. Right, and that happened in the 60s and the 70s, right? And there was this big bump up in finding into maybe the late 70s, early 80s. Big bump up in finding lots of new anti-cancer agents. So there have been little bumps along the way. It's just hopefully this strategy is a little bit more universal and gets at lots of things. And we're by no means solving, the problem's not solved, right? There's going to be lots of DNA that we pull out that we have no idea what to do with because we don't have good models for expressing that DNA, right? So we are getting at another portion of what's out there. We clearly aren't getting at everything that's out there. But the traditional approaches that tried to get around things where you knew there were limitations was go to a different environment, grow it in a different way, or put the same extracts in different assays in hopes of finding different molecules that function in different ways.
Just had another question. In terms of, I mean, you're one of the younger faculty, or you're youngish. And I guess basically the question I'm asking from a policy perspective, funding has been very tight. I wanted to hear your opinions or thoughts or suggestions about
Besides more money? You want something slightly more intelligent than that?
Especially from your point of view, from applying and trying to get money. It's been especially hard, I think, with younger investigators in general. And you're in a unique position because you're getting both money from public source and private source, and that private source certainly supposedly encourages high-risk research. But I just want to hear, anything that you've seen that you'd like to, suggest changing, or things that you think might be a good idea and you'd like to see implemented.
Yeah, it's what do you do without more money? I mean, it comes down to that, right? You either have to give more money or you have to figure out how to divide it up differently, right? And so I think those are your two options, right? And so my personal opinion, and part of it's because I'm young, is I think more money should be earmarked for young investigators, right? I mean, and I think that's, one is someone has to break the cycle of I can't complain. I've gotten my fair share of money since I've been at Rockefeller, right? I can't really complain too much about it. I have two grants from NIH, I have Hughes funding, I have Rockefeller funding, but it's actually, I would say that the difficult part is not the first 5 or 6 years, because there are some private foundations that fund those first years. You can receive a variety of awards. Your university funds those first years. There's actually some preference at NIH for giving early investigators a little bit of benefit of the doubt. It's that mid-career range where you get thrown in with everyone else, right, that I think hurts a lot of people, where you should have young groups growing because they have the longest time for their science to do something, as opposed to bigger groups growing bigger. So I think the only thing you can say is you either got to give more money or you have to figure out what's the better way to divide the pie. And I think divide the pie to a younger class of people that are mid-career, right, would be a more productive strategy for the money. Hopefully that doesn't go online, what else can you say? I mean, I think if you're going to say we have the amount of money we have, you have to figure out how you get it to people that can use it for a longer period of a career. Get their career started. I think that's what's happening now, is there aren't lots and lots of careers getting started, right? And there should be many, many more careers in science getting started early on with lots of funding available to get their ideas out there.
How do the compounds that you find are generally toxic versus having specific activity towards bacteria or fungi?
Yeah. So lots of the antibiotics are specific for bacteria. Lots of the anti-cancer agents are general cytotoxins. That's kind of how it ends up. But almost every anti-cancer agent you take, even successful ones, are general cytotoxins. Right? So that's, and we do have some compounds that we're looking at, which I didn't show you, that we're trying to chase the target down that don't kill bacteria but do kill human cells. And so they break down like that. But lots of anti-cancer agents, ones we report as anti-cancer agents, the ones that are actually used in the clinic as anti-cancer agents, are general cytotoxins. Lots of the antibiotics are just antibiotics. And so if you go to this slide, let's see. Just an antibiotic, toxin, right? Just a human cell line toxin, just an antibiotic. So they break down to those 3 classes, but yeah, whole range.
I'm going to tag on to something to Richard's question a little bit. So the reason why, or I'm assuming the reason why you've gotten the private funds that you have is because you're doing cutting edge, novel research, right? And I'm just wondering, in times of tight budget controls, would your research have been funded through the NIH, do you think? I know scientists don't like to second guess, but I just wonder if this research now would be funded.
Yeah, well, it did get funded. I got an NIH grant my first year at Rockefeller, so I can't say that it wouldn't have gotten funded.
But would you
I don't think it's
safer in your claims, or just basically
right.
Not make
Right, and part of it's a slightly different career. I mean, it's a new field and those kinds of things. I think what won't happen, or what is going to be difficult to happen, is now that we have, I think, cool results, it's going to be difficult to scale that. Right? I don't think we're. I think your first grant, to some extent, is you get lucky. I get lucky. I mean, I think it's a great idea. We had some progress, all those kinds of things. But I think a lot of people would argue that they put in a dozen grants and one of them gets funded, right? They just get lucky the first time. I think the real issue, and I think a lot of people would say that is everyone recognizes the first grant is difficult, but actually it's the subsequent grants that you have this good idea. How do you continue it, right? And because it's just a version of what you've done before, right? And so I think for me that's going to be the issue. Is I think we've got some cool results. How do we get funding to scale that? And I'm not sure that exists, right? So what do? We put in lots of grants this year, right? And takes a year to review. We'll see next year.
Like a database or something with the results that's publicly available to that other scientists?
Yeah, so that's what we're going to do. That's what this is going to be.
Okay. Oh, I see.
So the actual paper was just accepted, so that'll go live in about a week. And you'll be able to log on, and people will be able to say, I want molecule X. And they'll be able to see whether, now there's still a lot of work to be done once you find the genes that make molecule X. The question is, can you teach everyone to do that, right? And will we be able to get all those genes? But yeah, that's the idea behind this, is that, we'll keep some of it to ourselves that we want to work on, right? But the other stuff, there's lots of it out there. You'll just make it available. People will be able to type in that I want to find molecule X. My lab works on kinase inhibitors. I want to find more kinase inhibitors. And you have the name of the molecule, and it's a natural compound, so you have genes. You can search those genes against the database and figure out, I'm going to go into this well of the library and find that gene cluster that maybe makes new versions of those. So yeah, we're definitely going to do that.
Will it be interactive where other scientists can pick up soil samples and put their data on them?
That's so yeah. One of the reviewer comments was, what are you going to do with so we've written a piece of software that we're going to put on this server and people can do it. Are you going to make the software available for anyone to have? And our answer was no. And the rationale behind that is that what we want to force people to do is, if they want to use it, they should make their library available to other people to see it. See, it's unfair to make things freely available, everything available, because then you can't work on it. So they make available most of it, right? Because there's going to be so much sequencing that comes out of it that they can't work on it all. So work on the stuff you want, make everything else freely available. Because quite frankly, we're not going to make everything freely available, right? I mean, there's got to be something we can work on. But that's the idea of the software if you want to use it. And you can argue whether it's high tech or not, but it's a nice piece of software that allows you to do these comparisons. And if you want to put your sequences in there to run them through, then you have to make those publicly available and somehow try and make the clones available that would then allow someone else to look at it. That's somewhat of a pipe dream, right? That's what we'd like to do, right? It's a lot of infrastructure to do that costs money, right? And then developing a brand new system. Where do you get money for that? Is NIH going to give it? I don't know. I mean, we could try and write a grant for it, but that you've got to host a server. You have to have someone to recover clones. You have to do a lot. If it's really going to be a useful tool, it doesn't just come out of thin air. Right?
But we did it for GenBank, and we're doing it for a lot of the
But
proteins.
this is
I
true.
think this is where it's going.
So we'll see. That's what this is supposed to be. Snapped. So you can log on at some point in the next couple of weeks and find your favorite compound. And, come work in the lab getting that compound.
Thank you very much.
Well, thank you guys.